Okay, let's try talking about the urinary system. All right, we're going to go over the organs and talk about what they do. That's the high-level overview first part of this. Anyway, so the urinary system carries out the removing of waste from the body, okay? Most of these waste products are high in nitrogen. That's why urine is high in nitrogen. Urea is the main thing in urine. That's why it has its name. And as you probably guessed, it's nitrogenous. All right, so urea is made from breaking down amino acids to release, you release ammonia, very, very toxic stuff, bad stuff. The liver is going to combine CO2 with the ammonia to make urea, which is still not great, but a lot better. Makes sense. Makes sense. We say someone has uremia if they've got too much waste in their blood. This can screw with your heart. Vomiting, breathing problems. The reason why, the book isn't saying this here, but the reason why it would do some of that stuff, if you remember, muscles, how the muscles work, a lot of that depends on chemistry and ion concentrations, and you have to have the right amounts of the right ions. Well, you don't want other ions floating around in there and messing that up. The same thing. Respiration, the triggers, not the only triggers, but one of the main triggers for respiration is based on chemistry of the blood, carbonic acid and CO2, basically. And if you have other acidic type things happening, you can mess stuff up, and that can make it difficult to breathe. Anyway, let's move on. So creatine, you probably heard of creatine phosphate, yeah. Creatine phosphate's an energy molecule. We're breaking it down, and creatinine, yeah. Hold on a second, I'm going to check something. Okay, yeah, I was mispronouncing that. It's creatinine. Creatinine. Anyway, it's what happens when we break down creatine phosphate. So, uric acid, if you get too much of it in your blood, you'll get crystals that form in the joints. That's gout. That happens from eating a lot of rich foods, meaty foods. Anyway, your urinary system helps you maintain your water-salt balance. Basically, water's precious. We don't want to lose large amounts of it. We want to get the waste out, but we want to keep the water in if possible. So, and it's not mentioned right here, so it'll be mentioned later on, I'm sure. Water can diffuse across membranes, sort of, kind of, but the kidneys, water movement's a lot more important. And at the end of a functional unit of a kidney, a nephron, you have these collecting ducts that several nephrons feed into. They have the ability to make water channels called aquaporins that can greatly change the water balance. It's like a tube that water passes through in or out of a cell, or in or out of the filtrate that's turning into urine. Much better at this than trying to use simple diffusion. All right, sorry, simple osmosis. So let's move on. The main function of the kidneys is this water-salt balance. Salts like sodium chloride have the ability to influence the rate and direction of osmosis. Let me make this really clear. Osmosis is the movement of H2O. The O in osmosis should remind you it's the movement of water. And straight, simple, water wants to dilute things. That means water wants to follow salt. You'll probably hear me say that many times, that water wants to dilute stuff, water wants to follow salts. So if you want to get water to go somewhere, pump a bunch of salt there. Anyway, the more salts there are in the blood, the greater the blood volume and the greater the blood pressure, because the water goes there. All right. By regulating the concentration of sodium and potassium in the blood, the kidneys help to regulate blood pressure. And I'll give you an extreme example of that. If someone has a COPD, or sometimes just from having high blood pressure, a doctor might prescribe a diuretic. They're doing this to try and have you pee out the extra blood volume so the blood pressure drops. Anyway, kidneys also maintain appropriate levels of bicarbonate, calcium. That's all good. We've talked before about how bicarbonate is also influenced by respiratory rate with the lungs, but moving on. It says maintenance of a health, it says a healthy blood pH is about 7.4. That's accurate. That's the middle of the typical range. Your blood could honestly be as low as 7.35 or as high as 7.45. 7.4 is right there in the middle. Now, if your blood drops below 7.35, we're going to say, oh, this person has acidosis. Their blood is too acid. And the chemists the chemists who are here with us are going to be like, but 7.35 is still very slightly basic. Correct. But it's a lot more acidic than what we want it to be. So pH to be between 7.35 7.45 for blood. Blood will get into different conditions depending on what you've been doing. So you've been running really fast for a while and you haven't been able to get enough oxygen. Your muscles are going to generate lactic acid to keep going. That lactic acid in the interstitial fluid makes the fluid more acidic. And that means that your blood will be probably below 7.45. When the red blood cells get to that part of the muscle tissue with the low pH, the hemoglobin is going to be less fond of holding onto oxygen, more likely to dump oxygen. This is nice. This means that if you're running and you generate lactic acid because you can't keep up on respiration, well, you generated lactic acid. And the body can see that lactic acid. And because you are at this lower pH, the hemoglobin is going to drop the oxygen right where it's needed most. So you can turn that lactic acid back into pyruvate and continue with the conversion and getting all the energy out. Anyway. So kidneys monitor and control pH. They can reabsorb bicarbonate from the filtrate, or excrete hydrogen ions. This is why urine can be acidic. So, yes, you're a good dog. Urine can be slightly acidic, and that is the reason why people, I know this sounds gross, but there are people who believe, and they are wrong. They are absolutely wrong. I'm telling you, this is not the right choice. Who think that, oh no, I got stung by a jellyfish. I need my friends to pee on it. No. In reality, you need acid. You need any type of mild acid, okay? And yes, urine often is slightly acidic. That could be a thing. But, you know, I was out snorkeling with a group of students, and one of them got a jellyfish sting. I opened up my pack and I took out my lunch. And I mean, this is when we were back on shore. And I got out an orange and cut the orange up, and we squeezed orange juice on her arm. That's a lot more acidic than urine would be. Heck, Taco Bell mild sauce is vinegar. Okay, sorry, the number one ingredient or number two ingredient is vinegar. So yeah, put Taco Bell sauce on it. Go to Long John Silver's and keep little packets. Anybody I know who spends a lot of time around the ocean, they probably have a bottle of vinegar in their cooler anyway for jellyfish stings. Because you take out nice cool vinegar, that'll feel good from the cool anyway. But yeah, you put something acidic. Anyway, so back on topic. Kidneys can excrete hydrogen ions. That means kidneys can make urine that is acidic, that's removing hydrogen ions, that's removing acid from the blood, from the body. There are better sources of acid for jellyfish stings. So don't fall for that if it's not necessary. All right. Urine is slightly acidic because we eat foods that are acidic. pH will be 6 or a little lower. It's not going to ever be down stomach acid 2. That would be like eating your urethra with the acid. It would be bad. All right, so let's move on. Secretion of hormones. The kidneys can make hormones. Renin, this is part of the renin-angiotensin mechanism that we'll talk a little bit about. Aldosterone is made by the adrenal glands, which are inside the capsule of the kidney. They sit on top of the kidney proper. Obviously, the adrenal glands are most famous for making adrenaline, but aldosterone as well. And again, aldosterone is a water salt balance thing. The renin-angiotensin mechanism, that is too. The kidneys also, if you're low in oxygen, they release EPO, erythropoietin, and that causes red blood cells to mature more quickly, so you can get more red blood cells. So if you're low in oxygen, if your kidneys are screaming for oxygen, they're going to release EPO, and that'll cause you to have more red blood cells. You probably guessed that messing with EPO levels is one of the oldest methods, not the oldest, but one of the oldest methods of blood doping for athletes to try to have artificially high numbers of red blood cells. Hold on a second. Yeah. Okay. So I just went and found a link to, it's at an athletics website, UFC website actually, talking about EPO and blood doping, and I put it down there for you. I also put one up since we were just talking about the pH balance of urine and how people used to talk about it, and there are better sources of acidity like vinegar, and vinegar being used for jellyfish. I threw 2 links for how to treat jellyfish and how not to treat jellyfish. Put those in there too. Let's move on. Okay, other things the kidneys do. They can reabsorb some of the nutrients that get filtered out and could get into the urine. You like, oh no, that's important. Keep that, pull that back into the body. They can help you make vitamin D. Vitamin D promotes calcium uptake. Remember, we talked about this for the digestive system. Vitamin D is needed in order to take up calcium. Vitamin D is partially manufactured by your skin when you're exposed to UV light. But we spend a lot more time indoors. We're a lot more scared about skin cancer. A lot of people get vitamin D from their dairy products. You'll usually see your milk is fortified with vitamin D, for instance. But naturally, it's not just your skin. Naturally, your kidneys can help with making vitamin D. Anyway, let's move on. All right, so organs in the urinary system. I've got the kidneys, obviously, and we have the ureters, the urinary bladder, and the urethra. Those are in order from kidneys to the ureters, as the tubes to the bladder, to the urethra. That's the tube from the bladder to the outside. Now, if you get confused, how do I tell the difference of the ureters and urethra? Imagine that you have to go to the bathroom really badly. And when you do, you're going to say, ah. All right, well, if you keep that image in mind, and then you say urethra, like urethra, it's easy to remember. That's the one that carries urine from the bladder to outside the body. The ureter does not. It carries to the bladder, carries from the kidneys to the bladder. So it might help you to remember, like, oh, that felt good. I really needed to go to the bathroom. Anyway, moving on. So your kidneys, you have one on each side. They're partially covered by the rib cage. Due to the way that the liver sits, the left one is a little lower, not well protected. It's not saying this here in the book, but they're also retroperitoneal, which is a fancy way of saying they're behind the peritoneal cavity. You know, the hollow cavity inside the abdomen where, like, oh, your intestines are inside there. Well, the kidneys are not there. They're embedded in the body wall behind that. But anyway, the right kidney hangs a little lower than the left. They both are partially covered by the ribs. Bean shaped. That's why they call a kidney bean a kidney bean, because it looks like a little kidney. They're reddish brown in color, and they have a renal capsule around them. They also have cushioning fat pad, adipose tissue, and that's good since not fully covered, especially on the right. So. Yeah, right kidney lower than the left. That cushioning is good because you don't have the protection. Anyway, moving on. So concave side of the kidney. Concave, think a cave. You're going into a cave. That's the side that's like this, okay? The side like this is convex, all right? The concave side of the kidney is where the renal artery enters and the renal vein and the ureter come out. That's all true. The convex side would be the side towards the outside, the lateral, and the concave would be medial. I probably would have, if I was writing the book, gone with medial and lateral because that's easier. Anyway, the renal artery carries blood to the kidneys. The renal vein carries filtered blood away from the kidneys. So straightforward. The artery carries blood to the place it's named for. Now, ureters connect the kidneys to the bladder, just like the book says. There are 3 layers to the ureter. There's a tough outer layer, it's connective tissue. There's a muscle layer, and then on the inside you have this smooth, thin mucosa. It says peristaltic contractions in the ureter push urine into the bladder even when you're lying down. What are they talking about? It's the same as the digestive system. The muscle layer contracts with rhythmic circles around the, so here we have our tube. We're going to do, not with sound effects, but same idea. When you do this, it's going to push the thing through the tube. And that's how the ureter contracts. All right. So here we have our organs. Ureters going to the bladder. Going from the bladder to the urethra. Now, not mentioned here yet. I'm sure you'll see it eventually. In the male, that the ureter goes through the middle of the prostate gland. Think of the prostate gland as being like a donut, and there's a hole in the middle of the donut, and the ureter goes through it. And that's why if men have an enlarged prostate, you're going to have you could have narrowing of the urethra, and that makes it difficult to urinate. Anyway, but we'll get into that stuff later on, I'm sure. So, prostate. So I'm going to say this is a female they have drawn here. Anyway, so the urinary bladder stores urine. You might remember when we did histology, that's one of the few spots in the body where you see a lot of transitional epithelium. It changes shape as you go through the tissue layers, and it's really stretchy because of it. Also, they're talking about rugae, the stretch. Has 3 openings. The 2 ureters coming in and the 1 urethra coming out. There's a urethral sphincter there that lets you, a muscle ring that lets you control when you're going to release urine. So and then you go into the ureter. Hold on a second here. It talks about folds of the mucosa. Okay, so the PowerPoint was just about to talk about rugae, or folds that allow for expansion. And I just wanted to pull this picture of a cactus up for a visual so you could see it. You can see how the cactus, they've got these grooves. I mean, you can see them really well there. You can see them super well here. But barrel cactus have grooves. And whenever they want to expand, like they're absorbing all the water, there's a rain and they want all the water, they absorb it into their tissues, these folds expand out. And if every one of those expands out, what happens is that the whole thing gets bigger, fills up with water. Okay, well, your stomach does the same thing. Your stomach has folds that we call rugae. It's not absorbing, it's just filling up, like an accordion fills with air. And so it was this space inside the accordion. Then when it fills up, those accordion folds fill, it gets bigger, wider, bigger, wider. Same exact thing is happening. What happens to the cactus, what happens to your stomach, it happens to your bladder. You've got some folds, some rugae in it, and that allows the bladder to get bigger, to fill up with fluid. Anyway, I'm going to flip back to the PowerPoint now. Okay, so the bladder stores urine. It's got 2 openings for ureters, 1 for urethra coming out. I already told you if it's a man, that goes through a prostate gland. It's like, here's the bladder, prostate gland is right below it. Anyway, it's got 2 sphincters to control the flow of fluid. An internal sphincter, smooth muscle, you got no control over that. The external sphincter is skeletal muscle. That's what lets you pee on demand and not pee when you don't want to. So easy enough. But I wanted you to know that rugae, those folds, allow for expansion. Anyway, let's move on. There's a fancy name for urination. It's micturition. The urine fills with bladder. You get stretch receptors. They send signals to your nervous system, telling you that you need to urinate. The spinal cord can send signals down to the urinary bladder, telling it to contract and squeeze, and the sphincters to relax so that fluid pushes out. But obviously the external sphincter is under skeletal muscle control, voluntary. You can decide if you want to urinate or not. That lets you get to the urinal, get to the restroom, and take care of it. Anyway, so here's the picture of the bladder. You can see the little rugae, the folds on the inner lining there. As it fills up, the stretch receptors, they're going to send a signal to the spinal cord. and the spinal cord will go through interneuron, if the spinal cord signal through the interneuron to go down through the motor neuron to the external sphincter to let you release the urine. All right, let's move on. So the ureters took urine to the bladder. The urethra takes urine away from the bladder to the outside of the body. In the short female urethra, it makes it possible to get, it makes it easier to get infections, urinary infections. The male urethra is longer because it goes through the middle of the penis, and it has to go through the middle of the prostate before it can do that. We already said this, if the prostate gets bigger, it's going to restrict the flow of urine through the urethra. That's why people with enlarged prostates, typically old men, they often feel like they have to go to the bathroom all the time, but when they try, they can't. That's because their bladder is full, but it's really hard to get urine past the enlarged prostate. Anyway, let's move on. In females, the reproductive and urinary systems are not connected. In males, the urethra carries urine during urination and sperm during ejaculation. Totally true. Not mentioned so far. It's probably something that we'll cover in repro, but I'm going to say it now anyway. There is a problem with that. If you remember, the urine can be acidic. Not saying it always is, but because urine can be acidic and because sperm are easily killed by acidity, we are going to try and neutralize the acidity in the male reproductive system, in the urethra, before we pass sperm through. There are multiple glands involved, but one of them is the Cowper's gland or bulbourethral gland. That is at the base of the penis, and that produces pre-ejaculate, or pre-cum is what some people call it. But that pre-ejaculate is it's smooth, it's a thin mucus, it's smooth, slippery, and because it's basic, it will line the urethra prior to ejaculation, neutralize the acidity as it goes through, but also the lining is slippery, make it easy for the sperm to go through, nothing gets stuck behind, nothing gets killed. That's the plan. Anyway, so I figured when they're talking about ejaculation, I wanted you to know that we have glands to protect the sperm in the semen. I'm sure we'll talk more about the prostate gland and the seminal vesicles. Anyway, but moving on. So here's a check your progress. I'm going to pause for a second, and you can make sure you answer you're capable of answering all these. If you're not, go back and watch that section of the video again. Okay, let's move on. So we're going to talk about the structure of the kidney. Just like a lot of places in the body, the outside will be a cortex and the middle will be a medulla. We'll probably talk about the fact that it's divided into little lobes. It's divided into lobes. We have what we call renal pyramids. The pointy tip of the pyramid, regardless of the orientation it's in, because they change like that. The middle of, the tip of the pyramid will be called the papilla, meaning finger-like. The rounded part on the other end, the wide end, will be called the base. Have the arcuate arteries along the base. All of the papilla are aiming towards the medulla of the kidney. The pyramids are separated by renal columns. Each papilla will drip urine that is being formed into calyces. And we have minor calyces that only collect urine from, a minor calyx only collects from one renal pyramid. Major calyces collect from more than one, and those drain into the renal pelvis, which then drains into the ureter. Anyway, so let's move on. We'll go through and you'll see some of these things. So the outside is the cortex, the middle is the medulla. The M in middle should remind you of medulla. And the medulla has these cone-shaped renal pyramids. The renal pelvis is a space in the middle where the calyces empty into, and the renal pelvis drains into the ureter. All right, so here we have one. This pyramid's good and pointy and pyramid looking. Some of them are definitely not so much, but cortex for the outside, medulla for the middle, arcuate blood vessels, arcuate artery there, renal columns. Like I said, a minor calyx collects from one, a major calyx. I'm not pointing the blood vessels, I'm pointing space underneath that space. You can see it's collecting from this pyramid, this pyramid, this pyramid. That'd be a major calyx. And right here we have a renal pelvis connecting, sorry, this tube, when we are below these blood vessels, that would be the renal pelvis. And you can see all of the urine from all the pyramids is draining into that. All right, let's move on. So the functional unit of a kidney is a nephron. It's tiny, and I'm going to go through the parts right now. It will sound like a lot of parts, but it's not that bad, really. So we have this little knot of blood vessels called the glomerulus, and around that we have the Bowman's capsule. And the Bowman's capsule, we call it the filter membrane between the two, between the glomerulus capillaries. Now, and the glomerulus capillaries are interesting because it's one of the only places where you have arterial going in, arterial going out. Usually an arterial goes into a capillary bed, and then you have a venule that comes out. But here we have an arterial that goes in and an arterial that comes out. So the one that carries blood to the glomerulus, the one where blood arrives, is the afferent arterial. The one where blood exits is the efferent arterial. All right, so fine. Anyway, but glomerulus, Bowman's capsule. Then we have the proximal convoluted tubule. Convoluted because it wiggles a lot, moves all around. But proximal convoluted tubule is the one close to the glomerulus. And then we're going to have the loop of Henle, the descending and the ascending. And then we will have the distal convoluted tubule. And then we'll go into a collecting duct. And many nephrons, several nephrons will drain their urine into the collecting duct. It collects from more than one nephron, hence its name. Anyway, moving on. Okay, yeah, this is, nephrons are the functional unit. It has all that stuff I just described. Several nephrons drain into one collecting duct. Okay, you've seen that. All the collecting ducts empty into the renal pelvis. Actually, this is one pyramid, that's the papilla, and we are draining into this calyx, which drains into this renal pelvis. But that's being picky. If you looked under a microscope, you would see kind of a through that pyramid. So we have the base with the arc, and we have the papilla here. It's kind of a triangle shape going up. You guys see the lines. Those are the collecting ducts. Well, collecting ducts and loops of Henle. All right. Anyway, so blood supply to the nephron. When we come from the renal artery, we're going to have an afferent arterial. The A in afferent should remind you that blood is arriving at the glomerulus. The E in efferent Should remind you the blood is exiting the glomerulus. It is going away. The glomerulus has higher blood pressure than anywhere else in the body, simply because we have a wider pipe going in than we do coming out. Why do you want high pressure? So you're always pushing things through the filter membrane, okay, from the glomerulus into the Bowman's capsule. And now, the honest truth is you're going to reabsorb most of that stuff. But that's how we get it into the filtrate. Then we decide, do we want to pee it out or do we want to keep it in the body because it's precious? All right. You know, a lot of nutrients, you don't want to pee that stuff out. A lot of minerals. So the efferent arteriole is going to empty into the peritubular capillary network that surrounds the nephron. That's true, but I wish we had a picture. I bet we do. Okay. So when you see this, here's our glomerulus. And just like I said, that's going to empty into the Bowman's capsule. Blood will, the fluid in the blood will, like the blood plasma, that stuff will turn into what we call filtrate. At the proximal end of the nephron, that is filtrate. At the distal end down here, that's urine. Now, what changes along the way? We'll do some talking about it, but you're going to pass things in and pass things out. And the reason for this peritubular capillary network that they're talking about, all this purpley stuff, is as you are passing through, if you're like, that's a thing I want to keep in the body, you will remove it from the tubule, remove it from the nephron, put it into the interstitial space. And then that stuff will be absorbed into that capillary. And now you carry it off in the blood and you're good. You don't lose too much of whatever it is that you were worried about losing. Anyway, so these peritubular capillaries are surrounding the loop of Henle. And understand, we have a descending loop of Henle and an ascending loop of Henle. And the ascending and the descending, their membranes are structured differently. And it has a big effect on whether or not water can go across. We'll get into that. So the outer layer of this, we had our knot of capillaries. That was our glomerulus. And around that, the Bowman's capsule, also called the glomerular capsule. All right. The outer layer of that is squamous epithelial cells. No big deal. Small, they're thin. It's easy to reabsorb stuff if you want to. The inner layer are podocytes. The pod should remind you of podiatrist, a foot doctor, say. That they're foot cells is what that says. But what they are cells that have a lot of foot-like processes that come out and they cover the glomerulus. So you have the capillary, and then you have these foot cells that are like that. And between them, you have enough connective tissue to anchor the two together. Those three layers form the filtration membrane. And they will allow molecules from the blood to get out, but hopefully, if you haven't damaged it, your filtration membrane is going to keep really big things inside. So big proteins like albumin, not going to go through. Whole blood cells shouldn't be going through. If they do, you've got a damaged kidney. You've damaged your glomerular filtration membrane. Anyway, once we're through the once we're through that filtration membrane, we're in the capsule. The next thing we're going to do is move to the convoluted tubule. Convoluted just means twisty, and you can see this twist around, around. Oh, sorry, around. I will try doing it with my mouse so I don't point and switch it. This twisty, wiggly, that's why they call it a convoluted tubule. The proximal one is just close to the glomerulus. The distal convoluted tubule is just the other end away from the glomerulus. Now, this is a really popular drawing, the way it's drawn. Everybody has stuff like that in their anatomy books, their biology books. But in reality, imagine taking that the way it's drawn and now turning it around like this, making a circle, because that distal convoluted tubule actually is really close to the glomerulus. That's because we know that because we have these special cells, the renin-angiotensin mechanism we're about to talk about, and the JG cells, the juxtaglomerular cells. Juxta means like next to, so the juxtaglomerular apparatus. Basically, we're doing some last-minute chemistry checks on the filtrate before we throw it in the collecting duct, and it lets us know if we need to change what's being absorbed or not, what's getting into the urine. You're like, oh, no, we don't want to do that. You can do that for pressure. You can do it for composition. Anyway, let's move on. This proximal convoluted tubule, it's got cuboidal epithelial cells. All right. It has, in fact, that's the most popular thing. If we want to show someone simple cuboidal on a slide, we're going to grab kidney and show kidney tubules. But these tubules are covered in microvilli. Why? Imagine my fingers are the microvilli. There's lots of extra surface area. That makes it good for absorption, all that extra surface area. And if you have more surface, it's easier to absorb stuff. The idea here is that most of the stuff we put in the filtrate, we're going to change our minds to go, no, we don't want to pee that out. Let's absorb that back in. All right. So here we have proximal convoluted tubule. Here we have these awesome little cilia. They're increasing, the microvilli, they're increasing the surface area so we can do absorption. All right. Now the loop of Henle has a descending and an ascending, okay? The descending is to, in the descending, we're going to take salt out and put it into the medulla. Actually, let me, I want to double check that. But anyway, I'll just go with it. The idea is that as you come back up, water is going to try to move out of the tubule, and no, it's on the way down. On the way down, we're going down the loop of Henle, down into the medulla. As we do, that medullary space is filled with urea. It's up by the cortex. You got a little bit. As you go deeper, 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 it's more and more concentrated. So remember, water wants to dilute things. Water wants to follow salt. We're going to go down the loop of Henle. As the urea gets more concentrated in the fluids surrounding the tubule, water will leave the tubule, leave the descending, go out. Then as we turn and go on the up, suddenly we're like, nope, we're not, the membrane's not permeable to water anymore. What you just did is concentrated the urine. So anyway, let's move on. The distal convoluted tubule, you already saw, that's the one on the collecting duct side of the nephron, not on the glomerulus side of the nephron. All right. Its main job is exchanging ions between the blood and the renal tubule, the nephron. Okay. The distal convoluted tubules of several nephrons go into one collecting duct, and that collecting duct will carry urine. It says to the renal pelvis, but you already know it carries it to the papilla, where it drips into the calyx, where it goes into the major calyx, where it goes into the pelvis, where it goes into the ureter and goes into the bladder. But let's move on. Okay, so the glomerular capsule and the convoluted tubules are up in the cortex. The loop of Henle can go into the medulla, all right? And some of them go way down and some of them are pretty short, but basically the more you have to concentrate urine, the more you have to store water. Say you're a desert tortoise, your kidneys have long loops of Henle so that your urine is very concentrated. You absorb all the water, you keep the water because in the desert, water is precious. How concentrated is the urine of a desert tortoise? Comes out semi-solid, kind of like a paste. All right, and that's because they have long loops of Henle. So the collecting ducts are they're also in the medulla, and they're the things I told you that you have that striping look inside your renal pyramid. That's it. That's all the collecting ducts going towards the papilla where they drain out. So to check your progress, I'm going to pause a second. You see if you can answer these. If you can, great. If you can't, go back and watch this section again. All right, moving on. So we will talk about how we make urine now. We've talked about the parts. Let's talk about the actual making. And we'll talk about reabsorbing nutrients and salts. Like I said, we don't want to pee out anything precious. And we'll explain what we do get rid of through this tubular secretion, what we don't. So there are three stages, three steps in making urine. Glomerular filtration, that is stuff leaving the blood of the capillaries going into the tubule. Tubular reabsorption is we look at this filtrate that's going through and we're like, it has more salt than I want to lose. Let's reabsorb some salt. That type of thing. Tubular secretion would be, we still have too much of this. We need to get rid of some of that. Stick that in. Okay. So glomerular filtration is blood going into the glomerulus. It comes, you remember, from an afferent arteriole. Blood arrives through the afferent. And that's bigger than the efferent, so the pressure spikes, and that helps force fluid through the membrane from the capillary through the basement membrane through the podocytes. Anyway, the increased blood pressure will push more fluid out. Hi, Oscar, you're so helpful. We'll push more fluid out, and it will leave the glomerulus in bigger volumes because of the higher pressure than capillaries normally have. A lot of capillaries have leaky walls, but not like this. These are seriously leaky. All right. They're not mentioning it, but it also matters that these capillaries are what we call fenestrated. The German word der Fenster means the window. These are little windows all over them. These capillaries actually have holes like a strainer or a colander. So yeah, lots of fluid rushes out of them. All right. So the stuff, once the fluid has run out of the blood and it's in the nephron, we're calling it glomerular filtrate. This filtrate is what will eventually be urine after we take things out and add things to it and concentrate it and do all that. Then we call it urine. Okay, so the process is called filtration because big stuff and formed elements can't get through. We're talking about albumin and blood. Albumin and blood cells. The cells shouldn't be able to fit through. If they do, you've got kidney damage. The things like albumin shouldn't be able to fit through. Fibrin, fibrinogen, those things should be too big. So it says, in effect, blood in the glomerulus has two portions, the filtered stuff and the non-filtered stuff. Well, that's true, but you could say that about any blood anywhere. Oh, you've got the stuff that's big and you've got the stuff that's fluid. But all the big stuff gets left behind when we're doing this. All right. So here we have our nephron again with our glomerulus and our Bowman's capsule. Let's focus on this part right here. And pardon me getting this right up in my face. It's tiny on my screen, so. Okay, yeah, so the things it's showing leave the capillaries are water and urea, glucose, amino acids, uric acid, and salt. All right, but amino acids, yeah. An entire giant protein like an albumin, no, that's not what should be getting out. All right, let's move on. So what things are filterable can be, meaning what can we take out of the plasma? Water, nitrogenous wastes, that's urea, that's nitrates, nutrients, salts, yeah. Things that should not be in the blood, things that should be held back are blood cells and fragments. The fragments we call thrombocytes or platelets. Big plasma proteins like albumin, those things shouldn't fit through the filtration membrane. If they do, you have a very damaged membrane. Non-filterable stuff is carried out through the efferent arteriole. And let's see, filtrate has the same concentration of filterable components as plasma. Okay. Yeah, so the stuff that comes through should be about the same as the plasma because you're delivering a different volume of plasma coming out. All right, so tubular reabsorption. If we stick stuff into the tubule, we're going through proximal convoluted tubule, we're going to reabsorb some things. In other words, we're going to take them like, yeah, we put that into the filtrate. No, we don't want to pee it out. Let's keep it. We'll pull it out of the filtrate. We'll reabsorb it and put it back in the body. All right, so some of that happens passively. Some of it happens actively. We haven't talked before. When we did membrane transport, symporters go in the same direction. Antiporters go in opposite directions, okay? So a symporter, you're going to have things move in the same direction, say an amino acid and a something else, or a glucose and a something else. The little molecular pump made out of protein that's pumping things across the membrane, it's moving things in the same direction. Antiport, it moves them in opposite directions. So in this case, it says it's taking sodium into the cell while transporting hydrogen out of the cell, hydrogen ions out of the cell. Let me translate. The cell is part of the wall. Here's the cell, 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 the wall of the tubule. So if we are moving sodium into the cell, what we're doing is saying, sodium's a little too precious to pee that out. Let's keep that in the body, reabsorb it, take it from the filtrate in the middle, put it into one of the cells. Oh, and acid, yeah, the hydrogen ions, we got way too much of that stuff. Pump those into the filtrate and pee them out. All right, and again, this will regulate the pH of the body because if we move hydrogen ions into the urine and pee them away, we've just made the blood less acidic. All right. If sodium is moved, chloride will follow. So like if you actively pump sodium, positive charge, a negatively charged chloride is attracted to it, it will go with it. And this pumping, remember, was going from the inside of the filtrate to the cells that are around that form the wall of the tubule. What that means is that we are reabsorbing the salt. We're taking it from what would be peed out and put it up here in the cell where it's kept. About 65% of the sodium that gets into the filtrate is reabsorbed to keep in the body. So if you look right here, you might think, oh, wow, we pee a lot. Well, yeah, but we'd have to drink so much more to replace all the water if we didn't do what we do, which, look right here, we reabsorb 99% of it. Almost all the water that gets into the urine, we're like, keep that, keep that. So we have only a very small amount of water we need to replace each day. Anyway, urea, we don't reabsorb much of that at all because we're trying to get rid of that. We might reabsorb some of it to work on osmotic pressure, the concentration gradients and all, but we're trying to move that through. We're trying to get rid of it. All right, so glucose, amino acids, we do some reabsorption of that. We've got membrane proteins like, hey, that's an energy source. Don't just pee that out. Keep that, keep that. Diabetes mellitus. Mellitus means honey. That's because the urine is full of sugar. And back in the olden days, before they had good tests, so I'm not making this up, they would taste it. Like, oh yeah, that person has this disease. They have diabetes mellitus because they have sweet honey-like urine. I'm talking medieval times, but yeah, that's what they really did back in the day. What's happening is the glucose transporters that are supposed to reabsorb can't because there's so much glucose, and so it's being peed out. And because there's so much extra glucose in the filtrate that becomes urine, water wants to dilute stuff. There's more water in the urine too. And that means that people with diabetes mellitus have to pee a lot and they're thirstier than normal people. So I'll just read it straight from the book. The frequent urination and increased thirst experienced by people with untreated diabetes is due to less water being reabsorbed from the filtrate and into the blood. Why is less water being reabsorbed? Because the water is instead staying in the filtrate, trying to dilute the sugar molecules. All right, let's move on. Tubular secretion. We can move things from the blood to the tube into the filtrate on purpose, actively move them. You could do this with the acid, creatinine. See, I did it right this time. Drugs, that's why they talk about a half-life of drugs. You can do it with meds. This is going to happen along the nephron, all along the nephron, but it says in the end, the urine contains stuff that has been filtered but not reabsorbed, plus what you actively secrete into it. Urinalysis is analyzing, the process of analyzing urine. All right. Urine is usually pale yellow. The color depends on how concentrated it is. Colorless, dilute, that's better than, you're better hydrated if you can afford to pee out that much water. So dilute, colorless urine, dark, really concentrated, you need to be drinking more water. Things they look for in urine to see if they're abnormal: the amount of glucose you have, the ketones, which is related to breaking, ketone bodies are related to breaking down proteins. If you have too many ketone bodies, you would have ketosis. And because you're breaking down amino acids, you're making it acidic, making the blood acidic. Anyway, nitrates and nitrites are nitrogen compounds, as you probably guessed from their name. Blood in the urine, that should not exist. All right. White blood cells. Remember, all the blood cells should be staying in the blood, not in the filtrate. All right. So here's to check your progress, and go ahead and make sure you can answer all those questions. If you can't, then go back and rewatch this section. I'm going to pause for a minute and give Oscar some belly rubs, and then we will continue. That's enough. All right, let's move on. So we'll talk about the kidneys and homeostasis. Remember, homeostasis is maintaining internal body conditions. The most important ones here, if you think about them, are for the kidneys, are going to be water balance and acidity. So summarize how the kidney maintains your salt water balance. State the purpose of ADH. That's a hormone. The name is antidiuretic hormone. The hormone diuresis is peeing. You would normally pee out more water than you do, but ADH, antidiuretic hormone, will make you pee less. But we're going to talk about ADH and aldosterone and how they work. Explain how the kidneys assist in the maintenance of pH levels in the blood. We already said that if you get too acidic, you're going to pee out the hydrogens. If you're not acidic enough, you will pee less hydrogens out than normal. All right, so the liver is going to make urea, the muscles are going to make creatinine, and all the body cells make uric acid. These wastes are carried in the blood to the kidneys, where we filter them out. We also get rid of some urea across the skin. And if you have a kidney failure, you'll release a lot more urea across the skin, and it forms a frost-like layer. Anyway, moving on. So salt water balance, most of the water is reabsorbed. You remember it's 99% of it is reabsorbed. All along the nephron and the collecting duct, you are reabsorbing water. The reabsorption of salt always precedes the reabsorption of water, which occurs by osmosis. I know I'm reading that directly, but let's refer back to what I told you before. Water wants to dilute stuff. Water follows salt. So if we reabsorb salts, if we take salts out of the filtrate, here's the inside the tube, we take them out and put them into the cells around, then water will want to follow and it will leave the tube. And that will mean that the filtrate inside the tube gets more concentrated. All right. During reabsorption, water passes through water channels within the plasma membrane called aquaporins. And if you want to reabsorb more water, you can make more aquaporins. All right, more than 99% of the salt that's filtered at the glomerulus is returned to the blood. The kidneys will reabsorb other things. We're talking potassium, magnesium, bicarbonate, basically keep the precious things in the body. All right, so if we look at this, reabsorption of salt and water from the cortical portions of the nephron. So cortical portions, we're talking up in the cortex, not the medulla. All right. 65% of the water that's filtered in the glomerular capsule is reabsorbed in the proximal convoluted tubule. How do we do it? Like we just said on the previous slide. First, we absorb the sodium, the chloride follows, and then because we have aquaporins, water's like, oh, I need to go there. I need to dilute that stuff. Water goes through the aquaporins. It says hormones regulate reabsorption of sodium and water in the distal. So what we just described was proximal convoluted tubule. Now we're talking distal. So proximal is the stuff before the loop of Henle. Then we have the loop of Henle, and we have distal on the other end of the loop of Henle. Aldosterone is an adrenal hormone that it's going to promote ion exchange. What that means is that we are going to get rid of potassium and sodium will be reabsorbed. We'll keep the sodium. And aldosterone is released by your kidneys. Great. Ah, finally. Do we have a pretty picture? Yeah, we do. Do you remember I said that here's the normal drawing of a nephron with the glomerulus and the proximal convoluted tubule and the loop of Henle and the distal convoluted tubule and the collecting duct. And I said, but in reality, that whole thing is curved like this. So the distal is close to the glomerulus. That's what you're seeing here. Okay, so we have our glomerulus, our Bowman's capsule. We have our big fat afferent arteriole bringing blood, our little skinny efferent taking blood away. The higher pressure is forcing things through the filtration membrane between these pieces of podocytes into the tubule. Great. All right. But look right here, in between the afferent and the efferent, that's the distal tube. We've bent it around, and now afferent and efferent, right between them, we have the distal convoluted tubule. And that matters because even though they're not labeled here, we have the juxtaglomerular cells, JG cells in there. And this is the renin-angiotensin mechanism that I was talking about. All right. ADH, antidiuretic hormone. If we don't have it, we can't reabsorb in the distal. It's made up here in your hypothalamus, and it's given off by the pituitary gland, the backside of the pituitary gland. And that happens if the blood is getting too concentrated. Water will move from the distal tubule into the collecting duct back into the blood. It gets absorbed into the blood. All right. So ANH, atrial natriuretic peptide, this is special. We don't normally think of the heart as an endocrine thing, but watch this or read this. A hormone secreted by the atria of the heart when they're stretched due to increased blood volume. If you've got too much blood pressure, usually because you got too much blood volume, the heart screams. It's like, no, too much. And it releases ANH, which causes you to get rid of more sodium, to do less reabsorption of the sodium. So sodium goes out in the urine. Water wants to follow salt. More water goes out in the urine. The blood volume goes down, which means the pressure goes down, which means the heart says, ah, thank you. All right, so ANH causes water to be released. Here we have the same picture we had at the beginning. We were talking about different organ systems. Let's move on. It's just, hey, this is what this system does. All right, we get to talk about the loop of Henle. They're calling it loop of the nephron. I will always call it loop of Henle. That's the way I learned it. Sorry. We have an ascending and a descending. The descending is first, and it goes down into the medulla, and then we turn the corner and go back up. And it says salt passively diffuses out of the lower portion of the ascending. Yeah, but talk about the descending, because we do the descending first. Water is going down we're going down the descending as the filtrate is being pumped down the descending loop. Okay. It says countercurrent mechanism, osmotic gradient in the medulla. You've got lots of urea down where it's concentrated in the medulla. And so as you go deeper, deeper, deeper into the medullary cavity, you have more water, more water, more water leave the tubule and go into the surrounding tissues of the kidney, the medullary tissues. Then we go around the corner. We're going to go up. We're going to change the membrane. Now water can't go across anymore. Okay. So let's see. Water leaves the descending limb on its entire length, blah, blah, blah. Salt passively diffuses out of the lower portion of the ascending. Why? Well, we're going from concentrated in the medulla, we're going up, 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 up. The surrounding stuff is less concentrated, less concentrated, less concentrated. And thus it's very salty here. So the salt's very constant, it's getting more and more and more concentrated relative to what's around. And so the salt's like, oh, I want to move out there. Just by changing whether water can cross the membrane or salt can cross the membrane, we remove water and then remove salt, and we get rid of a huge amount of the urine with that loop, a huge amount of the water in the filtrate before we turn it into urine, just by having that loop of Henle. So the fluid in the descending loop encounters increasing osmotic concentration of the solute. Like I said, it's urea, and it's getting more and more and more urea around the tube. So water leaves the tube and goes into the surrounding tissue, because water wants to dilute stuff. Water wants to follow salts. The ascending can't absorb water. So you lose water. Water could go either way. Water could go out, water could go in, water could go out, water could go in. But because we're going down, it's getting more and more and more concentrated, water goes out, because water wants to dilute stuff. Then we go for ascending, and we don't have any aquaporins, and water can't come back in as easily. So a lot of water went out when we went down. We go up, the water is gone. It's not in the urine, and it can't move back easily. But salts can move out. The ascending, it says the job is to establish a concentration gradient. The water is returned to the cardiovascular system. That's pretty straightforward. So there you see on the descending side of the loop of Henle, the nephron loop, we lose water. On the ascending, we don't allow water to go out, but salts do. Anyway, moving on. So the collecting duct, you already know it collects from more than one nephron. That's why when you look here, you have one there, one there, one there, one here. Each collecting duct, multiple nephrons. And you got that same osmotic gradient as you're going down, 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 down. So water wants to go across. It says water diffuses from the entire length of the collecting duct into the blood if ADH is present. In other words, if you're doing the antidiuretic hormone thing and you are making lots of aquaporins, yeah, great. Diuresis is increasing urine. Antidiuresis is decreasing the amount of urine. Straightforward. ADH will make you reabsorb water instead of pee in it. And so you have less water volume in your urine. You have less urine. All right. Now let's talk about blood pressure. We did this a little bit when we talked about the atrial natriuretic hormone, ANH, but if your blood pressure is low, exchange doesn't happen, and you can't do glomerular filtration, this is a problem. So you'll, if you have decreased sodium in the blood, it's an issue. So what happens? What causes it? Maybe you do a lot of sweating. Your blood pressure falls, and renin-angiotensin starts. The kidneys will then do more reabsorption, and they'll keep as much salt, as much water as they can. All right, that's straightforward, not scary. When sodium is reabsorbed from food, the sodium content of the blood is higher than normal. I said reabsorbed, I meant absorbed. When sodium is absorbed from food, we've got more salt in the blood than normal. This means that we'll have more blood volume than normal because the water wants to follow. That means that the atria is stretching. The heart will then release ANH, atrial natriuretic hormone, which will stop you from reabsorbing as much sodium and water, and you'll increase your urine volume. You'll decrease the blood volume, increase the urine volume, pee it out, get rid of the extra salt and water. Diuretics are meds that they give people to increase the amount of urine that they're peeing out. It doesn't have to be. The example they used first was alcohol. If you ever notice someone's in a bar and they drink, and in a little while they have to go to the bathroom. Well, that's because alcohol stops you from making ADH, stops you from making antidiuretics. If you don't make antidiuretic hormone, you're going to diurese more than normal. You're going to pee more than normal. Caffeine increases glomerular filtration, decreases tubular reabsorption. That's going to, if you're decreasing reabsorption of sodium, like here we have our tube, we're doing less reabsorption of sodium across. That means more sodium stays inside. Water wants to dilute things, water wants to follow salt. We have more water on the inside, so we're making more urine. Diuretic drugs for high blood pressure will lower the amount of water reabsorption, decrease blood volume and pressure. We're talking about things like LASIK. Makes you pee all the time. All right, so the normal pH for body fluids is 7.35 to 7.45, like our blood. This is the pH where proteins have the right shape, enzymes, which are proteins, work properly. If you move above or below that range, they start to denature, take on weird shapes that don't work real well. Alkalosis is what we call it if your blood is more basic, above 7.45. Acidosis is what we call it if your blood is more acid than normal. Now you might say to yourself, but that's not acid at all. It's not even neutral. Neutral is 7 and it's above that. Yeah, all true. But it's closer to being acid than it normally is. It's below the normal amount, so we say it is in acidosis, even if it's only at neutral or slightly above. We eat a lot of foods that are acid or base, and when we do, that's going to change the composition of the blood temporarily. The way we keep the body fluids, the pH in our body fluids the way it is, we could use the acid-base buffering system. That's based on bicarbonate and carbonic acid. The respiratory center, that is based on how much you breathe, how quickly you breathe, how deeply you breathe. Basically, if you're not, you can, without even thinking about it, your body can make you more or less acidic in the blood on purpose. Subconsciously, you don't know it's happening. How? Well, if your body makes you breathe a little harder, a little faster, you're going to be moving more carbon dioxide than normal, getting carbon dioxide out of the body, which means you're getting rid of carbonic acid, which means you are counteracting the acidity in the body. Like, hey, the body's too acidic. Let's breathe a little harder, a little faster, and we'll literally blow out some of the stuff that's making acid. You can go the other direction. If your body's too basic, if your blood is too basic, your body can start to breathe a little slower, a little shallower, and you're going to build up carbon dioxide in the lungs. And that carbon dioxide will then enter the blood and be used to make carbonic acid. And that'll bring the pH back down a little. All right, so buffers are things that keep you in a constant pH range. Doesn't get too high, doesn't get too low. what buffers do is reabsorb hydrogen or hydroxide, and they can also, some of them can release it. So we already said that carbonic acid and bicarbonate are the two biggies. So I said that on the previous slide. There you see it. All right. Oh, there it is. How beautiful. All right. If hydrogen is entering the blood, then it's going to combine with the bicarbonate. It's going to make carbonic acid. You might say, oh, but doesn't that make it more acidic? No. Remember, pH is a measure of the amount of free hydrogens. If we have a bunch of free hydrogens here, and we do something that takes them out of the hydrogen and makes them part of a chemical, you reduce the amount of hydrogen. You reduce the amount of acid. Now, OH works the opposite. The more OH, hydroxide, that you have, the more basic it is. So you might say, oh, we've got this and we've got carbonic acid. Well, this carbonic acid can combine with the hydroxide, and now you're making water and carbonate. Let me point this out another way. Here we have the bicarbonate, here we have the bicarbonate, here we have the carbonic acid, here we have the carbonic acid. These buffers can work in a reversible reaction, go in either direction as needed. I should do it like this, to keep the pH from going too high or too low. It keeps it in the range we want it, which for blood is 7.35 to 7.45. All right, let's move on. Yeah, a blood buffer can be overwhelmed. Basically, if you put in more acid than you have bicarbonates to absorb it, the blood will get acidic. The next adjustment to keep the pH occurs in the lungs. There's the complete blood buffering system as I normally draw it out. You can see that right here, if you breathe hard and blow out carbon dioxide, you're going to use up, because this is reversible, has to reach equilibrium. If you get rid of carbon dioxide, then carbonic acid will break down to level out to bring the carbon dioxide back up. But if that's true, you're going to make more bicarbonate, which means you're going to absorb these two things, hook them together. What that means is that you have less acidity in the blood because you have less free hydrogens. All right, you will, if you are needing to get rid of hydrogens, get rid of acids, you will breathe harder, breathe deeper, breathe faster, and do it without even thinking about it, because your body's trying to get rid of the hydrogens. The kidneys don't react as quickly as the lungs, but they can do a lot more. They can absorb bicarbonate, they can get rid of hydrogens, we can pump them into the urine to make it more acidic. So if the blood is really acidic, we're going to take hydrogens out of the blood. We're going to take bicarbonates and reabsorb them. So ammonia is another way of removing hydrogens. If we take ammonia NH3 and combine it with a hydrogen, we can pee that out, and that took hydrogens out of the blood. So here we have pictures of the kidney tubule next to the capillary. Understand that generally speaking, we're going to take bicarbonate out of the kidney tubule, put it in the blood. We're going to take hydrogen out of the capillary and put it into the tubule. That means that we're going to make the blood less acidic. All right. Kidneys are assisted by other systems. Oh, sorry. Assist other systems. We already talked about how the kidneys, when they feel like they're low in oxygen, they make EPO, erythropoietin, and that causes red blood cells to be produced and grow up faster in the bone marrow. The kidneys control how much calcium is in the blood. This is stuff that you check for in the filtrate. They can convert vitamin D to its active form, which makes it easier to absorb more calcium. So if the kidney's like, there's not enough calcium, you can make vitamin D to make it easier to get that calcium. All right. They can regulate the sodium and potassium content. We've talked about that too. That's pretty straightforward. Because you do so much reabsorption in the proximal convoluted tubule, you could cut that back if you want to pee some sodium out, or you could do it a little better if you want to keep some sodium in. All right, here's a check your progress. I'm going to pause this. You can read them over, make sure you answer them. Make sure you can answer them. If you can't, rewatch this section and then try answering again. I'm going to pause for a second, and then we can move on. Okay, let's move on to the very last section of this chapter, chapter 11. So we're going to talk about kidney diseases and disorders, major problems. Talk about hemodialysis, kidney dialysis. You've heard people talk about it. Although now there are forms that'll let people walk around and stuff. And talk about kidney transplants. All right. So disorders of the kidneys. Lots of, remember, it's a high pressure system, filtration system. So diabetes, where you're pumping a lot of urine through. Hypertension, where you have high blood pressure. Those often come side by side, so doubly bad. They can cause disease. They can cause the kidney to be progressively damaged more and more over time. Remember, itis is an inflammation, so urethritis is inflamed, infected urethra. Cystitis, think of a cistern, a water chamber. That's your bladder. So cystitis would be infection of the bladder, urinary bladder. Pyelonephritis would be an infection of the kidney itself. You're infecting the nephrons. All right. Urinary tract infections, enlarged prostate, messed up pH, any of that, you could get kidney stones. They're mostly calcium phosphate, uric acid protein. And if you're lucky, they're little ones and they pass without you even knowing it. If they're big, they could block the renal pelvis, block a ureter. Pressure will build up in the kidney because you're not draining it. And it'll destroy nephrons. And that's bad. If that happens, you can't remove waste from the blood, and waste builds up in the blood. Anyway, you probably know about lithotropy. They can take this. Lithotropy, they can take a person and put them in a tank of water and put sound in, aiming it at the kidney stone, and pulverize the kidney stone to break it up. Now, that's not, it's not as simple, oh yeah, they could just break it up. No, those pieces are going to have jagged edges, and those jagged edges are going to scrape and scratch. I've never had this done, but I know someone who did. And they said that passing a kidney stone or pieces of kidney stone, the fragments, really hurt. Anyway, but let's move on. One of the first signs of nephron damage, if the pressure destroys the filtration membrane, you're going to start getting parts of blood that normally are not in the the urine, they're going to be there. So you'll see albumin, that's the main protein in blood plasma. You'll see white blood cells, maybe even red blood cells. None of that should be in urine. And urinalysis, analyzing the urine, you can detect that. If you damage the kidneys enough, urea and other things are going to stay in the blood instead of being filtered out. And if 2/3 of the nephrons don't even work, we call that uremia. If you're not removing salts, not removing water from the urine like you should because you've damaged the nephron, you're going to have edema. You're going to have fluid accumulating, puffy ankles, puffy legs. That you might say, oh, well, who cares? Well, it can mess up your ion balance. It could stop your heart. It can kill you. So if you've got damaged kidneys, you're probably going to be on dialysis. And they'll take a dialysis machine. They'll take blood out of your body and pass it through tubing, dialysis tubing. And this is artificial tubing that has really tiny pores. Only little things can get through, big things can't. So just like a normal filtration membrane, the blood cells, the albumin, stuff like that should stay behind. Sodium should be able to escape. Sodium should pass through. It's an artificial kidney machine, basically, and you pass patients' blood through the dialysis tubing, and little things leave in the machine and are hauled away and removed from the body. Big things stay behind. All right. Substances that are more concentrated in the blood are going to move. In other words, what they're saying, none of this is active. This is all passive. So it's all diffusion. Things that are more common in the blood, higher concentration in the blood, they're going to move into the tissues. Things that are more concentrated in the tissue, they're going to move into the dialysate, the fluid in the dialysis machine. And things in the dialysis machine, if you put them at higher concentration, they could move into the blood going through the dialysis tubing. Anyway, the old way this was done, you'd have to be hooked up to a machine a couple times a week and sit there for they say 3 to 6 hours as the stuff was removed from your blood, poisons were removed from your blood. We now have ambulatory versions. We can remove bad things from the blood. They collect in the peritoneal cavity, and then we drain them out. This allows you to walk around, have a somewhat normal life. You're still going to be sucking fluid out of the person's body 4 to 8 hours. But at least they don't have to sit there in a chair the whole time. If you have kidney failure, you could have a kidney transplant. Obviously, any organ transplant, there's a risk of rejection. Currently, the survival rate is 97% if it's a close relative's kidney, 90% if it's not a relative, but they think you have a good match. The holy grail that they're shooting for, and I actually know someone who's working on this project. There are people who are in Florida, up at Shands, who are trying to take animal kidneys, pig kidneys, wash the pig cells out. I know that sounds weird, but remember, just like a lot of things, like your skeleton, the cells are not the whole thing. There's a lot of protein, a lot of a protein scaffold that's left behind. If you wash the cells out, then we'll take that scaffold and put human stem cells on and grow a human kidney. So they could use stem cells from the person who needs a kidney and grow them a new kidney to transplant in. That's the ultimate goal. We're years away from that. The person I know is a microtome technician. That's a fancy way of saying she's taking really thin slices of the artificial I guess you call it an artificial kidney. But artificial implies it's not biological. A better way to say it would be these human pig hybrid kidneys. She's taking thin slices of them and looking at them under a microscope, preparing them so they can be looked at under the microscope. And the thing is, we got to make sure all the cells are growing the right way in the right direction, all the different kinds of cells. But if they can get that down, they could have transplantable organs and end organ shortages. It would be astonishing. Anyway, oh wow, and that's the very end. So check your answers. I might find a link and try to put it below about the pigs and the kidneys, and check it out.